Hello students, welcome to EPG Pathshala. I am Dr. Nilofer Khan from DPAS DRDO Delhi. Today I am going to talk about membrane potential, transmembrane potential and its measurement by microelectrode from the paper Membrane Biophysics. And the objective of the talk are understanding the membrane potential, how to calculate membrane potential, to understand how the membrane potential is generated, and the contribution of sodium-potassium ATPase, transmembrane potential, resting potential, action potential, and measurement of transmembrane potential by microelectrode. It is not possible to understand membrane potential and transmembrane potential without understanding of the structure and properties of the biological membrane. So I want to give some background about it. The membrane potential plays an important role across multiple scientific disciplines such as physiology, biology, and chemistry. In 1902, Bernstein, a German physiologist, came up with a hypothesis for how neurons work. His hypothesis basically stated that the resting potential of a nerve cell was due to concentration potential of potassium ion. Professor Bernstein hypothesized that there were three contributing factors to the membrane potential, namely membrane permeability and the difference in the potassium ion concentration, higher inside and lower on the outer side of the membrane. The second one was Walter H. Nernst. He was also a German physicist who is well known for the development of the Nernst equation and won 1920 Nobel Prize in Chemistry. He was a major contributor to the study of membrane potential. Nernst helped establish the modern field of physical chemistry and contributed to the electrochemistry, thermodynamics, and solid-state physics. He developed the Nernst equation to solve for the equilibrium potential for a specific ion. Next, Goldman, Hodgkin and Katz furthered the study of the membrane potential by developing the Goldman-Hodgkin-Katz equation to account for any ion that might permeate the membrane and affect its potential. The study of membrane potential utilizes electrochemistry and physiology to formulate a conclusive idea of how the charges are separated across a membrane. Now, coming to the introduction, what is a cell? It's a fundamental, basic, functional and biological unit of all non-living organisms. Cells consist of cytoplasm enclosed within the membrane, which contains many biomolecules. All living cells maintain a potential difference across their membrane. And what is the membrane? The membrane surrounds the cell. It separates the interior of the cell from the outside environment. It is composed of lipid and proteins. It is semi-permeable. And the potential difference, that is also called membrane potential, this is due to difference in concentration and permeability of sodium and potassium on either side of the membrane. Structure of the membrane. There are also built-in macromolecules of proteins and with various functions. This figure is showing the structure of plasma membranes. It is composed of phospholipid and proteins. It is about 35 angstrom, and the chemical composition of the plasma membrane, as I told you, 50% proteins, 43% cholesterol or phospholipid, 4% other lipids and 3% carbohydrates. And the capacitance of the membrane is about 1 microfarad per centimeter square. Resistance is 10 to the power 9 ohms centimeter square. Several types of proteins are embedded in the membrane. There are peripheral membrane proteins. There are integral membrane proteins. They can be penetrating or non-penetrating. Penetrating means they are also called transmembrane proteins. Like ion channels are transmembrane proteins which carry out the transport of ion from one side of the membrane to the other side of the membrane. Understanding membrane potential. The membrane potential is created due to various proteins embedded within the cell membrane as well as the structure of the phospholipid bilayer. As I told you before, the membrane proteins are of two types: peripheral membrane proteins and integral membrane protein. Integral membrane proteins are ion channels which conduct the transport of ion from one side of the membrane to the other side of the membrane. The cell membrane, it is an electrically non-conducting thin bilayer of about 6 to 8 nanometer. It is made up of phospholipid molecules, as I told you before. The phospholipid molecules, they have polar head and non-polar tails. The membrane that surrounds a cell is composed of phospholipid, glycolipid, cholesterol and proteins, as I told you in the previous slide. This picture is showing lipid bilayer. They have phosphate or polar heads and non-polar tail. The polar heads are hydrophilic and the hydrophobic tails are non-polar. Function of the biological membrane. The cell membrane perform many functions, and few of them are: cell membrane work as a barrier between the inner and outer surface of a cell. The membrane structure regulates entry of ions and exit of ions to determine the concentration of the specific ion inside the cell. They keep constant chemical composition inside bounded areas by selective transport mechanisms, and their specific structure and selective ion permeability is the basis for bioelectric phenomena. Membrane potential and transmembrane potential. All living cells maintain a potential difference across their membrane. Membrane potential is also called as Vm, voltage of the membrane, is a voltage difference between the intracellular and the extracellular fluids. Vm is due to the difference in the concentration and the permeability of ion on either side of the membrane. Vm is always negative inside relative to outside. It is measured in the unit of volts, and Vm ranges from -40 millivolts to -200 millivolts depending upon the cell types. Generation of membrane potential. Membrane potential in the cells are determined primarily by three factors. 1. The concentration of ion on either side of the membrane. Number 2. The permeability of the cell membrane to those ions through specific ion channels, and by the activity of electrogenic pump, for example, sodium-potassium pump and calcium transport pumps. Important points: how does it work? The difference between the electrical and chemical gradient is important. What is electrical gradient? Electrical gradient opposites the chemical gradient. It represents the difference in the electrical charge across the membrane. And the chemical gradient, it opposes the electrical gradient. It represents a difference in the concentration of a specific ion across the cell membrane. The Nernst equation can help us relate the numerical values of concentration to the electrical gradient. There are leak channels in the membrane. These channels are always open. They permit unregulated flow of ions down an electrochemical gradient. Sodium-potassium ATPase actively transports sodium ions out of the cell and potassium ions into the cell. It helps to maintain the concentration gradient and to encounter the leak channels. Ions and the resting potential. What are ions? Ions are electrically charged molecules, for example, sodium ions, potassium ions, calcium ions, and chloride ions. The resting potential exists because ions are concentrated on different sides of the membrane, intercellular side and extracellular side. Sodium and chloride ions are more outside the cell. Potassium and organic anions inside the cell. The major contributions usually come from sodium and chloride ions, which have high concentrations in the extracellular side, as I told you before, and the potassium ions, which along with large proteins, anions have high concentration in the intercellular side of the membrane. In the figure you can see. Maintaining the resting potential. Sodium ions are actively transported. Active transport means this uses energy to maintain the resting potential. Sodium-potassium pump It is a membrane protein or transmembrane protein exchanges 3 sodium ions for 2 potassium ions. How to calculate membrane potential of a cell? The membrane potential can be calculated. For the calculation, we use Nernst potential or Nernst equation. The calculation for the charge of an ion across the membrane, Nernst potential, is relatively easy to calculate. The equilibrium potential is calculated using Nernst equation, which is given below. E ion, E is the potential, I for the ion to be calculated, for which the concentration or the potential to be calculated, and where RT by F is approximately 61. Therefore, the equation can be rewritten as below. Derived under resting membrane condition, when the work required to move an ion across the membrane, meaning up its concentration gradient, equals the electrical work required to move an ion against a voltage gradient. In the above equations, R is a gas constant, T is temperature in Kelvin, F is Faraday constant, Z is the electrical charge of an ion. It can be +1 like for monovalent cations or +2 for magnesium and calcium. I out is the ion concentration outside the membrane or cell. I in is the ion concentration inside the cell. Membrane potential. A modification of Nernst equation is the Goldman-Hodgkin equation. It can be used to predict VM or membrane potential when the membrane is permeable to multiple ions. The only difference between Nernst equation and the Goldman-Hodgkin-Katz equation is that it adds together the concentration of all permeable ions as given below. This equation is used to calculate the membrane potential resulting from all the participating ions when VM or membrane potential is not changing or at resting state. Px, the relative permeability of the membrane of X ion measured in centimeter per second, an ion's contribution to membrane potential is proportional to its ability to cross the membrane. The permeability ratio of potassium to sodium to chloride is 1:0.4:0.45 at rest. If Vm is known, it can be predicted direction of movement of any ion. Resting membrane potential. All cells have resting membrane potential. The cell membranes maintain a small voltage or potential across the membrane in its resting state. In the resting state, the inside of the cell membrane is negative with respect to outside. This voltage arises from the differences in the concentration of sodium and potassium ions across the cell membrane. The membrane potential of a cell derives ultimately from two factors. First one is electrical force and the second one is diffusion. The basis of membrane potential. The electrical force arises from the mutual attraction between particles with opposite electrical charges, that those are positive and negative, and the mutual repulsion between the particles with the same type of charge, both positive or both negative. In the selectively permeable cell membranes are ion channels which allow potassium ions to pass into the interior of the cell but block sodium ions. Negatively charged proteins on the interior of the cells are also denied passage across the membrane. Active transport mechanisms are mainly sodium-potassium ATPase provides an additional contribution to the resting potential. Next is diffusion. Diffusion arises from the tendency of particles to redistribute from the regions where they are highly concentrated to the regions where the concentration is low. The collective action of these mechanisms leaves the interior of the cell membrane about -70 mV with respect to outside. Sodium-potassium pump, it is also known as sodium-potassium ATPase. The specific example of active transport is sodium-potassium ATPase. The pump is transmembrane protein. It is in charge of generating a gradient of ions. For each ATP that is broken down, it moves 3 sodium out and 2 potassium ion in. It uses about 30% of the ATP energy available to any cell. It is called ATPase because it uses energy in the form of ATP breakdown. Sodium-potassium pump. This figure is showing how this pump works. Membrane protein or this sodium-potassium pump is opened from inside the membrane where the three sodium ions come and they bind to the membrane inside of the protein, and then ATP molecule also bind. In the next step, the sodium ions are transported outside the membrane and 2 potassium ion bonds to the outside of the protein. The next step, potassium ions are transported into the cells and 1 ATP is used for the whole process. Sodium-potassium pump and its mechanism. The process of transfer of sodium and potassium by the following mechanisms. Enzyme linked to ATP or membrane protein and the 3 sodium ions from the inner side of the membrane, and the degradation of ATP occurs. This leads to phosphorylation of the enzyme or the transporter protein where the stems of ADP groups and the remaining phosphate are connected, and at this point no change in the spatial structure of the enzyme or protein and lead this change to the transfer of the sodium ions from the interior of the cell membrane facing the cytoplasm to the outside of the membrane. Transmembrane potential. Transmembrane potential is the difference in electrical potential across the cell membrane. There are a number of factors which contribute to this difference. First one is the differences in the ion concentration across the cell membrane. Main ions are sodium and potassium. Sodium ion concentration is high outside the cell as I told you before, and potassium ions are more in the intercellular side of the cell. Second one is the cell membrane selective permeability, means some molecule can get through the membrane easily and some need some protein or carrier to get through them. That is selective permeability of the membrane. Potassium ions are more permeable than sodium ions. Proteins are too large to cross the membrane. As a result of the above factors, the cytoplasm tends to be negatively charged. The extracellular side is positively charged. The resistance of the lipid bilayer to a free flow of substances across the cell membrane helps to maintain this potential difference across the membrane. Resting potential. As I told you, all the cells have resting membrane potential. This is inside of the cell has negative voltage with respect to outside of the cell. Under resting condition, this is called the resting membrane potential. A resting cell tends to show a fairly stable transmembrane potential across the membrane. With appropriate stimulation of the cell, this negative membrane potential may transiently become positive owing to the generation of action potential in neuronal cells or neurons. The resting membrane potential varies according to cell types. There are two types of cells, excitable cells and non-excitable cells. For excitable cells like nerves and muscles, they have higher potential than other cells. Those are non-excitable cells like epithelial cells and connective tissue cells. Its values depend upon the type of the cell. There are few examples given with their resting membrane potential. The action potential. The action potential is generated by the activation or opening of the sodium channels and subsequent inactivation of voltage-gated sodium channels with a slight delay in the opening of voltage-gated potassium channels. Sodium-potassium pump, although the immediate basis of the action potential is the activation of voltage-gated sodium and potassium channel, long-term support of the action potential requires sodium-potassium ATPase to maintain the concentration gradient. The action potential is a rapid depolarization of the cell membrane. It starts at the axon hillock and passes quickly along the axon. The membrane is quickly repolarized to allow subsequent firing. It is always the same size. In this slide, I am talking about the neuronal action potential. Microelectrodes. What is an electrode? An electrode of very fine caliber consisting usually of a very fine wire or a glass and filled with saline or a metal such as gallium or indium, that is white melted, used in physiological experiment to stimulate or to record action currents of extracellular and intercellular region. A very small electrode often used to study electrical characteristics of living cells and tissues. Single unit recording provides a method of measuring the electrophysiological responses of single neuron using a microelectrode. When neuronal action potential is generated, the signal propagates down the neuron as a current which flows in and out of the cell through excitable membrane regions in the soma and axon. The microelectrodes can be carefully placed within or close to the cell membrane, allowing the ability to record intracellularly or extracellularly. Microelectrodes. Microelectrodes are used for different type of recordings. Naming, first one is potential recording, second current injection, third introduction into the cell of ion selective resins for measuring potential or determining the free concentration of cytosolic constituents. Potential recording and current injections are the procedures underlying conventional microelectrode recording like voltage clamp and patch clamping. Microelectrode or ME might be an electrode constructed with a tip having the dimensions of a order of a micron, maybe 1 micron, 2 micron or so on. Usually this means a glass micropipette which is filled with an electrolyte solution to act as a conductor of electricity. Microelectrode making. While recording with the microelectrodes, it is very important how electrodes are made. They are made up of glass capillaries and there are different procedures by which they can be made. Glass microelectrodes are made by heating a capillary until molten. When it is stretched, while the glass is still plastic but cooling down, the step draws out, breaks and separate. And in one step, two microelectrodes are made, and they can be made by different processes, namely by hand. This is not recommended because the lack of reproducibility, because every time tip is not same. Secondly, it can be made with the help of vertical puller. This usually has a nichrome filaments and a two-stage pull. The first pull is by gravity and the second is electromagnetic. The third one is horizontal puller. These pullers are gear driven and have a platinum foil heating element. They are good for fine tips, but this type of pullers produces rather long wispy shanks. It is very important how electrodes are pulled and what type of tip we are going to have with what type of puller. Second type of horizontal puller, these pullers have usually have a platinum or nichrome heating element with a range of preheat times and 1 or 2 stage pull. They have good reproducibility, can be achieved with this puller. They are generally good for fine and short shanks. There are two types of shanks, long and short. They have different meaning for different type of recording because different type of tips and shank length are required for different type of recordings. Microelectrode filling. Because tip is so small, it is not easy to fill microelectrodes. So glass microelectrodes are filled with a salt solution. The composition of the solution can be determined by the individual experimenter and depends on the experimental protocol. In recent years, the preferred method of filling is to pull electrode from the capillary that has a glass fiber fused into lumen. When the ME is pulled, the lumen shape is preserved up to the tip. Using fiber containing capillary, MEs can be backfilled with a small amount of solution. The solution tracks down the channel formed either side of the fiber right down the tip. After filling the tip, they can be filled with the help of the syringe or other methods. But they should be filled halfway and there should not be any bubble. If any bubble persists, there may not be any contact at all, or if contact is there, they can produce large amount of noise if any bubble remains there. Microelectrodes, mechanical stability. Mechanical stability is very important when you advance the microelectrode into the cell or on the surface of the cell. The interaction between microelectrode and the cell needs to be stable on a submicron scale to avoid damage to the cell membrane and consequent excess current leakage. There are two symptoms of the mechanical problems: slow drift of the microelectrode tip with respect to the cell and faster vibrations. Micromanipulators should provide precision of the movement less than 1 millimeter with stability in the same range. This is achieved by either lightweight design moving short distance mounted close to the cell on the microscope or stage, or by more substantial units mounted on the base plate. Measurement of transmembrane potential by microelectrode. So, transmembrane potential can be measured with the help of microelectrode. It is measured with a glass micropipette filled with solution which conduct charge. The micropipette is inserted through the membrane into the cell. The voltmeter measures the difference in an electrical charge between 2 points. In other words, the potential difference, it is measured in millivolt, as you can see in the figure on your right. Potential difference between the microelectrode inside the cell, that is negative potential, and the surface electrode outside the cell, that is zero potential, is equal to membrane voltage or membrane potential. The resting membrane potential is measured on a relative scale. The reference electrode is placed in the extracellular fluid. The extracellular fluid is designated as the ground and assigned a charge of 0 millivolt. In reality, the extracellular fluid is not neutral and has an excess of positive charge that balances the excessive of negative charge in the cell. The resting membrane potential is between minus 40 to minus 90 millivolts in nerve and muscle cells. Microelectrodes are also called, or they can be called as patch pipette, or these are also called pipettes. Best example of recording of membrane potential or other uses of microelectrode is patch clamp technique. In patch clamp, technology was developed by Irwin Neher and Bert Sekman in late 1970s and early 80s. It is the most famous for its revelation that current across biological membrane really does flow through discrete channels. For this work, they received the Nobel Prize in Physiology or Medicine in 1991, and the basic principle of this technology, the base principle of the method is to isolate a patch of membrane or piece of membrane electrically from external solution and to record current flowing into the patch. This is achieved by pressing a fine polished glass pipette which has been filled a suitable electrolyte solution against the surface of the cell and applying gentle suction. It is very important for this type of recording, the type of glass, the type and the shape of the patch pipette or microelectrode and their filling. If they are not pulled properly and their tip is not polished properly, there will be very difficult to record the current by these microelectrodes. Below is given the picture of Irwin Neher and Bert Sekman, and on the right you can see the patch pipette is attached to the cell membrane and one ion channel in the patch pipette. Because using these microelectrodes we can record even current from a single channel, and the size varies from this micropipette varies from 1 micron to 10 micron. Patch clamp technique, its type. To measure what's happening in or on a single living cells, scientists use a technique called patch clamp, which requires an extremely fine pipette held tightly against the cell membrane. This is an electrophysiological technique and gold standard technique in electrophysiology, and there are several configurations of this technique, mainly whole cell recording. The current from the whole number of channels present on the cell membrane can be recorded. Cell attached patch, the current can be recorded from a piece of membrane that is attached to the patch. Inside out patch, they are also known as excise patches, and if the size is bigger, more than 2 or 3 micron, giant patches can be made, and it can be outside out patch. For different type of recording, different tip of the micropipette or microelectrode is required. Each of the 4 different patch clamp configuration has properties that are useful for studying different kinds of problem. Recording from cell attached patches imposes very little perturbation on the cell under study. It allows single channel currents. Nearly all of the known types of ion channels can be resolved by this technique. A cell-free configuration, inside out or outside out, have similar high current resolution and provide control over the ionic milieu on the both sides of the membrane. Summary. So in summary, we can say that the membrane potential or membrane voltage is the difference in electrical potential between the interior and the exterior of a biological cell. This membrane potential is due to difference in the concentration and permeability of sodium and potassium on either side of the membrane. While this phenomena is present in all cells, it is especially important in muscle and nerve cells because changes in their membrane potentials are used to code and transmit information or signals. Not all the cell types have the same membrane potential, and the membrane potential can be measured with the help of glass microelectrodes. Thank you.